Welcome all the dignitaries present today in this prestigious lecture. Once again, we are here together on this platform. I welcome our honorable respected Vice Chancellor, Sir, Professor Vaidya P.K. Prajapati, Sir. You have always blessings upon us to carry on such type of proceedings, and time and again he insists us to organize such type of activities. Sir, I would like to apprise you that this is the lecture series being conducted along with JCKIC and IIT on technology-based evidences for Ayurveda in precision health. Sir, this series is continuous since last January 22, and so far we have organized 4 lectures, sir. First was on the 15th of January last year by Professor Metalliji on Ayurvedic for Precision Medicine, Introduction to Basic Framework. And then it was the next lecture on April 7th, 22 by Professor Ajay Agarwalji of Department of Electrical Engineering. That was on micro nanosensors for sensory perception based Ayurvedic diagnosis. And that third one was on 19th July by Ravi Vankaila ji on theranostic medicines, translation from bench to bedside beyond. And then the last one, since last one lecture was organized on 8th of September 22 by Professor Manoj Chaudhary ji of Electrical Engineering in IIT as emerging technology trends. It's very good to know that now Professor Manoj Chaudhary ji has been appointed as Vice Chancellor in Gati Shakti University at Vadodara. It's a very proud to inform all of you. And today we are here once again with Dr. Neha Jain. She is assistant from Biosciences and Bioengineering Department in IIT Jodhpur. And today she will be delivering the lecture on role of bacterial biofilms in medical science. To start, before I would like to congratulate, I'm proud to inform you that we are having a collaboration with IIT, and we are working upon a research project that is IoTech, and this IoTech center now has been established. The building of the IoTech center has been established in IIT, and recently on 9th of on 12th of May, our Honorable Secretary Ayush has inaugurated the building of this center, sir. And that was a very proud moment that our Secretary sir were here, Director Professor Shantanu Chaudhary was there, and our advisor Dr. Manoj Nesri ji were also there in the gracious presence with our Honorable Vice Chancellor, sir. And sir, we are heading towards the next lecture in this lecture series. I welcome you with wholehearted welcome, sir, and to all the dignitaries, all the lecturers, professors who are present here across India to listen this very viable lecture, because as far as this collaboration is concerned, this is basically bridging the gap between the modern science and the Ayurveda medical science, and that we are organizing through this lecture series. And today a very specific lecture on role of bacterial biofilms in medical science will continue on. I would like to request our Honorable Vice Chancellor, sir, to bless before we start this lecture. Respected Honorable Vice Chancellor, sir.
Thank you, Dr. Rakesh ji. This is the platform which has been given to all the scholars and all the teachers of the Ayurveda, and along with some modern scholars also. Today, Dr. Neha Jain, Assistant Professor, Department of Bioscience and Bioengineering, is going to deliver a lecture on role on bacterial biofilms in medical science. At present day, we have found that a lot of medical science and Ayurveda science, though everything has been found described in our Ayurvedic sciences. But how to understand the role in present context important point of view. Considering the point, Dr. Rakesh ji is organizing this very important lecture, role of bacterial biofilms in medical sciences. I have visited personally on this particular department, and we found that a very good startups are also coming to explain the science in the form of evidences. So I think this technology-based evidence for Ayurveda solutions emphasizing precision of health is very fruitful for us and our scholars to explain the um, principles in context of the modern technology. So I congratulate to Dr. Rakesh for arranging this type of the lecture series on the platform of the university for the benefit of our scholars and our science. So thank you. Uh, because I am outside the college, so I welcome Dr. Neha Jain for his lecture. Thank you. Thank you.
Thank you, sir. Thank you, sir, for your kind blessings. Now I start with the lecture today, sir. Before starting the lecture, I would like to introduce Dr. Neha Jain among all of you. Dr. Neha Jain has done his BSc and MSc from University of Rajasthan in microbiology, and then she did his PhD in from IISER Bohali on molecular biophysics. Then she did her postdoctoral from University of Michigan on microbiology. She has the research areas and interests in amyloids and neurodegenerative diseases and bacterial amyloids and biofilms. She's having 4 ongoing projects, out of which she is in 2 projects she is a principal investigator, and in 2 she is the investigator, co-PI, and she has 8 top rated research articles in prestigious journals, research journals, and she has been awarded by finalist BBA Rising Awards Award 2023, International Mobility Grant Award 2022, IPRO Collaborative Grant 2022, SERB Earlier Career Research Award 2019, ASM IUSSTF Visiting Research Professorship 2019, and EMBO Travel Award 2019. This way she has earned totally 6 awards so far, and so far 5 PhD and 3 MTech students are undergoing research in her supervision. So I now request Dr. Neha Jain to please start your presentation. Thank you very much, Dr. Neha Jain ji. Please.
Good morning, everyone. Thank you, Dr. Rakesh, for this kind introduction. I hope I am audible to all of you.
Yes, you are audible, ma'am. Awesome.
Thank you, Dr. Prajapati, for allowing us to present our work here and all the dignitaries present in this platform. I would like to discuss the work that we are doing in our lab and how we can collaborate with Ayurveda to bring out more solutions, including the technology. So with your permission, can I share the screen?
Sure, ma'am. Sure.
Thank you. I hope my screen is visible.
Yes, visible. Yes, visible.
So today what I'm going to do is I'm going to give you a brief overview of what are bacterial biofilms, why they are important, why it's important to study them in the disease condition as well as in the non-disease condition. And if we understand them, we will be able to cure most of the disease which are not directly linked with the bacteria. So just to, I hope there are some students also. So I just want to give you a perspective that biofilms are not new. The area is not new. It's been very, very old. So for anybody who is a layman, the earth looks like this. But for people like me who are studying microbiology for almost 20 years, the earth looks like this. So earth is basically covered with all the microbes. We have many more times microbes as compared to human beings on the planet. And now it has been recognized that the microbes might be present in other planets as well. So the question is that how these microbes are surviving throughout so many years, and they're still continuing to survive. So this arises long time back when the earth started to make these living forms. So initially, bacteria used to live in a singular form. However, due to environmental changes, it was a struggle for them to live as a separate identities. So that's why the bacteria decided to move into a multi-species adaptation. So they were single-celled, however, they came together to make this multi-species. And what they found is that if they can make a community, they will be able to survive much better. So this is a very common saying in the microbiology field that in a biofilm, a single cell combines with other cell to become a better single. So I'm going to show you that how biofilm community is advantageous to bacteria which are living inside them. So first, let's see what are biofilms. So these biofilms are basically, they are made by bacteria. Bacteria reproduces very quickly. So when it reproduces very quickly, it has different types of mutations that can happen in it. So it can have harmful mutations that will kill the cells. It will have helpful that will help to succumb the new advantageous conditions. Neutral, which will not accumulate. So this way, the bacteria is able to adapt to the threat quickly. And when they adapt to the threat quickly, they're able to survive more. Biofilm formation is one of the biggest adaptations for bacteria to survive under harsh environmental conditions. So this is a question that how some bacteria hide from antibiotics. The antibiotic hiding is because of the formation of biofilms or a bacterial community. So biofilm can be understood as we human beings live in a community. We have our houses, we have windows. Exactly in the same manner, these bacteria live in the community. They share the resources, they make their house. So the house which is present outside or the covering which is present outside is called as extrapolymeric substance. And inside this extrapolymeric substance, a lot of bacterial cell resides. The factors that govern the bacterial biofilm formation is mostly the environment in which they are present. In this biofilm community, there is a heterogeneous expression of the genes that contribute to the plasticity and flexibility of biofilm. What does this mean is that the biofilm, the cells inside the biofilms, they are much better as compared to the free living cells in that particular environment. Therefore, these communities, they protect the cells from environmental stress such as desiccation and antimicrobials. These biofilms help in invasion during infection, which I'm going to talk in a little bit later part, and they also induce the host immune responses. Where do we find these biofilms? So if you just think about, there is no place where you cannot find these biofilms. So even in the space, the spacecrafts that now we are sending to different planets, those also have biofilms. So just to give you a perspective, that biofilms can be present on a well pump. They can be present in the Yellowstone National Park. The Yellowstone National Park is very famous because the temperature is very high over there. So you would assume that there will be no bacteria or microbes. However, this yellowish color that you see is because of the biofilm formation. In human beings, you can find them in catheter. Catheters are basically the medical implants that go into human body. So these are the lot of biofilm cells that you see here. Even you can find the biofilms on the contact lens that we use. So no matter how much do we clean our contact lens, there are chances that you will ultimately get the biofilms. So these are the almost the external environments where we can find the biofilms. However, also related to human beings in a variety of infections. And once I show you, you will be surprised to see that there is no place in human body where you can't have a biofilm infection. Recently, it has been found out in 2022 that even the brain has biofilms from bacteria. So if you think about device-related infections, no matter where your device are, these all are vulnerable to biofilm formation. If we talk about tissues, all the tissues that are present in the human body, they are vulnerable, they are prone to biofilm formation. So basically, there is no place where you cannot form biofilms. And once these biofilms are formed, it is very difficult to remove them. And more importantly, these biofilms crosstalk with each other. So if they are formed in one place, they can always move to the other place and infect the other area. So the question is how they are formed and why they are so resilient. So the biofilms are formed when the cells sense a clue from environmental condition that it is not suitable for me to live as a single cell and float around here and there. So that time, the mobile cells or the motile cells, they decide to stick on the surface. This phase is reversible, so they will attach, they will go out, then they will attach again. And finally, with the help of cilia or villi, they firmly adhere to the surface. Once they adhere, they start to multiply. Once they start to multiply, they also start to secrete certain substances that makes a wall around them. That is where the maturation of biofilms takes place. And once this mature biofilms are formed, it's very difficult for us to remove them. So if you think about this, this is like a very protective layer to protect something inside. This outer layer is called as extrapolymeric substance or extracellular matrix. So these cells stay inside the biofilms for a very long period of time, unless and until they realize that the outer environment is favorable for them. Then they start to disperse and then they propagate to other place. So this way they can move from one place to another. And that's how they infect the different types of the tissues or the organs. Now, during this process, it's not just that the cells are adhering, they're staying and they're moving. They are doing a lot of stuff. What they are doing is when they're adhering to this, so the cells that you see here, if you follow the cursor, these are human cells, these are human epithelial cells. So once the bacteria starts to adhere, the cell starts to deform. Once the cells start to deform, there are a lot of immune responses that get activated. This burden of biofilm on the cell is not going to take it. So that's why another set of immune system is activated. From this biofilm, a lot of substances are released, which causes infections or which causes toxicity inside the human body. All this biofilm matrix attribute to. So the biofilm basically, it provides the architecture to the bacterial cell. It provides stability to the cells. It allows to make pores and channels so that the nutrients can go inside, the waste material can go outside. So it also acts as a measure for waste disposal and to localize the nutrient, because once you have to stay in a compact place, you want to make sure that the nutrients, you have enough of them and you can survive longer in that particular area, even if there is a nutrient scarcity. Ultimately, it forms a skin. So like we have a skin which protects us from many environmental insults. This matrix helps it in protecting from environmental insults as well. What biofilm gives to a bacterial cell? It allows to tolerate to many other environmental clues, and it also allows to retain the water. It helps in nutrient capture and it circulates all the nutrients and whatever waste is. So it acts as a mini digestive system. Although bacteria doesn't have a digestive system, but when they make a colony, the entire colony acts like a digestive system where you can absorb the nutrient, you can take the nutrients and then break them. Because of all these attributes, the biofilms, they act as a fortress, a very strong fortress, which is very difficult to remove. They increase the tolerance of the cells which are present inside, and they allow horizontal gene transfer. So this horizontal gene transfer is going to play a very important role once we look into. So this horizontal gene transfer is mostly responsible for antimicrobial resistance, which I'll be talking in next couple of slides. Quorum sensing is another very important aspect of biofilms. So quorum sensing is like voting in India. Quorum sensing is basically if you have few people, the vote doesn't count. But if you have too many people to vote for a particular party, then the vote counts. So this is exactly what happens in quorum sensing, that if you have less number of bacteria secrete certain molecules, these are called as autoinducers, but they get diluted in the media. Okay, so in the low concentration, you have an individual behavior. However, when the bacterial cell number increases in the biofilm, you have too many cells to produce a lot of inducers, and then the bacterial cell can sense that there is somebody near to me with whom I can cross talk. So it shows a group behavior. And these autoinducers basically become very problematic for human beings because these autoinducers, they act as toxins. So bacteria secrete these autoinducers, which maybe activate immune system or activate another arm of the toxicity. So quorum sensing is one mechanism by which one bacterial cell can talk to each other, and when they talk to each other, they decide to cause the harm to the human body. Major aspect for biofilms is the antimicrobial resistance or antibiotic resistance. Antibiotic resistance due to biofilm has become a global burden. WHO has said that by 2050, this AMR due to biofilm will preside the neurodegenerative disease as well, because whatever antibiotics we are designing or we are making, they are not working since the bacteria is living in this biofilm. So how do we get this antimicrobial resistance because of biofilm? So there are two basic things that we need to understand is that when a bacteria is living a community life, there are two different types of cells. There are persister cells, they are the sleeping cells. So they are resting cells, they are metabolically not active. Some of the cells which are at the periphery, they are active cells. So when we throw antibiotics to these biofilms, the persister cells, they are not going to wake up. So they remain alive, but they are not affected by antibiotics. Only few of the surface active cells, they get killed by the antibiotics. However, the number is much more higher for the persister cells. The another way by which the biofilm protects this protects these microbial drugs is by efflux pump. So you have a certain pumps through which you do not allow the antibiotics to get inside. The extracellular matrix, it acts as a shield to enter the antibiotics. If even antibiotics enter inside, the number of cells which are present in the biofilm is way too high as compared to the concentration of drug. So the drug becomes a limiting factor and it is not able to act on it. Sometimes the cells which are active, they produce something which binds to the your antibiotic and nullifies the effect of the antibiotic. More importantly, these cells, these active cells might have plasmids which confers resistance to the antibiotics. And due to horizontal gene transfer, that gene can be transferred from this cell to this cell and then to this cell. So overall, the cells gain resistance to antibiotics. So this becomes a huge problem. And if you look into any kind of infections, especially the hospital-acquired infections, all the bacteria, they form biofilms, and these biofilms become to most of the antibiotics that are given by the doctor. Now, this next important thing is that so far we were talking about that if you have a bacteria, you get these biofilms, okay, and in certain specific regions. But bacteria is also moving in the biofilms. So this is the indirect link of biofilms to these cells. So this is an example where we have our gut epithelial cells. We have bacteria which are making biofilms. So under normal conditions in human gut, these biofilms are being made by E. coli and other Enterobacteriaceae species in the gut. However, these bacterial biofilms, they are dissociated. They are not harmful for us. But during gut dysbiosis, when there is a shift between the microbiota, some of the bacterial species, they start to make biofilm. And due to this leaky gut, the bacteria or the biofilms or the biofilm components, they travel through either the gut-brain axis or the bloodstream. Once they cross this axis or once they enter the bloodstream, the whole scenario changes. Now we have to deal these bacteria in our brain system or in the bloodstream. Okay, so then in the gut-brain axis, what has been found that these bacterial biofilms or the component of these biofilms, they actually can cross-seed with the human proteins and they lead to Parkinson's or Alzheimer's disease. So there are a lot of papers which suggest that your Parkinson's or Alzheimer's actually start from the gut. So the very early symptoms are constipation and discomfort in the gut. And then maybe after 10-20 years, you realize that you start to suffer from Parkinson's disease. So we think that this could be one reason that the biofilm components, they might be traveling, they might be talking to the elements of the brain cells, which lead to neurodegenerative disease. And it also leads to neuroinflammation. On the other part, when these components, they are released in the blood, they activate certain immune complexes such as toll-like receptors. So the eDNA or extracellular DNA, it binds to the receptor and then it activates the immune system to secrete interferons. Interferons are the proteins which are secreted when you have an infection. So these interferons or the antibodies, they are secreted in a very high amount, which leads to autoimmune disorders. So autoimmune disorders like reactive arthritis or SLE, they were initially considered as sterile because there were no bacteria or microorganisms were found to be associated. However, people realized that the signals that you get in these autoimmune disorders could be indirectly due to biofilms. So now this is a very new branch to understand that how the disease, which are not directly linked to bacteria, we are going to connect it to microbes or bacteria, and can we identify the initiation of disease at a very early stage. This is one of the focus of my group at IIT Jodhpur, and I'll be talking a little bit more about in details. So now let's see that what are the properties, what are the structure of the outer layer of the biofilms. So as I mentioned that this is the extracellular matrix which is present outside. It is made up of three main things. One is exopolysaccharide. There are protein polymers which are called as amyloids, and you have extracellular DNA. So if we can target specifically to these components of the wall of biofilms, we might be able to tackle the problem of biofilms. As of now, the solution that we have is not target specific. And that is what my lab is trying to do, that how do we give specificity to target this? As I mentioned, that these are the two main components. Curla is a protein fiber. So my lab works mostly on these kind of proteins which form the bacterial biofilms. Do they make curli or amyloid? So this is a bacterial biofilm which is grown in the lab. So this is a Bacillus biofilm. If you look into, this is E. coli. So this, if you look closer, this is an E. coli cell which has these lot of fibers on the cell surface. And if you look it carefully, these are the amyloid fibrils. So if you remove these fibrils by mechanical force or by some chemical treatment, the bacteria is not going to make the biofilms inside the cells.
Okay.
So we can target the amyloid fibrils in the bacterial biofilms. And amyloid fibril targeting has become a new area where, can you hear me properly? Am I audible?
Yes, ma'am.
Okay.
Yes, ma'am.
Yes, you are audible. Yes.
Sure. So amyloid fibrils are one of the areas where people are trying to target these so that we don't get biofilms. Okay? So just to let you know that the properties of amyloids, these are the protein polymers, which are cross beta sheet structures. They stack among each other to give rigidity. So the amyloids in biofilms provide strength, provide resistance and virulence to biofilms. Coli is one example. This is amyloid, which is known as curli. And if you see these two cells stick with each other because of the formation of the curli. The beauty of this protein is that you can actually isolate this protein and work in the lab. So this is a purified CSGA isolated in our lab, and you can monitor the formation of amyloids. So now you have a handle to understand in the lab conditions that if you have this protein, how this protein is making amyloids under the lab condition, and under the biofilm condition. And if you deplete this protein, what is going to be the consequence in the E. coli biofilm formation? Okay. These are the different ways by which the biofilms can be disrupted. So the existing techniques involves the physical and mechanical breakdown or using the antimicrobial agent. However, both of them have certain drawbacks. The alternative techniques could be to use protein targets, to use antimicrobial peptides, or to use small molecules like Ayurvedic preparations to specifically target the biofilm components different ways. Okay? So how can you inhibit the biofilm formation? We can start from the beginning. We can inhibit the adhesion. So I mentioned that the cell has to adhere. So if you can prevent that adhesion, or you can interfere with the quorum sensing molecules, or you reduce the cyclic DGMP concentration, which is required for the movement of the molecules from inside to outside. So this is the way by which we can stop making the biofilms. And there are a lot of, I'll show you that there are certain reports which says that some of the Ayurvedic compounds, they actually interfere with the quorum sensing molecules. Another option to control is that if you have biofilms which are already formed, how you are going to remove them. So how to disrupt these preformed biofilm. And this is again an area of intense research, because even if you are disrupting the preformed biofilm, you are still having the cells. So then you need certain antibiotics to move it from one cell to another cell. In our lab is that we characterize anti-amyloid and anti-biofilm agents. The agents are of variety of types. So we use human proteins, we use nanorods, we use carbon dots, and we also use certain Ayurvedic preparations. So this becomes the target specific rather than a random selection. We identify the mechanism, how these agents are working, and then we would like to test them in the animal model. So basically we start with the basic science, and the study is designed such that you identify the targets, you understand the mechanism, so that you can lead to a translational product at the end of the day. So what are the strategies that we do? So you can use nanoparticles, human proteins, Ayurvedic preparations, small molecules, or peptides. We usually use human proteins, and I will tell you why human proteins are better, and certain Ayurvedic preparations are also better as compared to these preparations. So how do we work in the lab? So we target the components of biofilms, specifically amyloids, using fluorescence spectroscopy, biochemical techniques, microscopy, cell culture-based techniques. The idea of using these strategies and is to understand that can we design antimicrobial or anti-amyloid therapeutics? Can we find the role of microbial amyloids in non-infectious disease and identify the amyloid components in biofilms other than E. coli or bacillus? How it is important? So it is important because it will allow us to understand the link to find a cure. So this is an overview of how we work. So we think that the bacterial biofilms are formed inside the cell surface. These amyloids, which are the part of bacterial biofilms, they can, even if the cells are not translocating, these amyloids, they translocate themselves. They cross the blood-brain barrier, they reach to the brain, or they interact with the certain cells at the epithelial level. They lead to the Parkinson's or Alzheimer's disease. Through the inflammatory response, they also contribute to autoimmune diseases. And these bacterial functional amyloids, they can also be involved in other diseases such as cystic fibrosis, colorectal cancer, urinary tract infection. So the urinary tract infections in females is non-curable, mostly because of the biofilm formation. And if you look into those biofilms, this amyloids are the major component in them. And it is now also linked to the AIDS, that the HIV protein, it also cross-linked with the bacterial amyloids and the diabetic foot ulcer. So you can think about any disease and you will find that, oh, there is some connection with the bacterial biofilms over there. These were the things that I was talking about earlier, that when we went back and we looked into that, do we have certain things already there in the literature? So I could find certain herbal extracts and Ayurvedic formulations to inhibit the biofilm. However, how are studies different from these ones? And how the Ayurveda doctors can help us to find a better solution. So most of these studies, they are not targeted studies. So they are disrupting the biofilm, but they do not know the mechanism. And if we don't know the mechanism, we will not be able to connect it to any signaling pathways or any mechanistic details we will be able to get. So what we do is we do a targeted approach. So we have identified certain proteins that can act as biofilm inhibitors. And the basis of identifying these proteins was to use a protein which is present already in the E. coli that prevent the biofilm formation. So initially, what we do is we characterize the anti-amyloid activity of a particular protein in the lab. We have standardized assays. So this is basically a fluorescence assay. So it's basically a fluorophore. It binds to the amyloids. So if your amyloid is there, the fluorescence will be high. If you add that protein, you don't have amyloids, there will be no fluorescence. So once we confirm that a protein is inhibiting amyloid or bacterial amyloid, then we check the anti-biofilm activity of that particular protein. So ideally, in the presence of that protein, the bacteria should not be making biofilms. So here is an example. This is a transthyretin protein, which is responsible for transportation of thyroxine. This is, I'm just showing my postdoc work where we identified that transthyretin is a potent inhibitor of bacterial biofilm, E. coli. And this is the first report to show that a human protein can actually inhibit bacterial biofilms. So people have not studied this before. So if you see these are TM images that the cells have these fibrils. However, when you treat the cells with this protein, you don't get these fibrils. And if you don't get these fibrils, you don't get any biofilm forms. So this was a very revolutionary study that I did it during my postdoc. And this opened a lot of doors that can we design the peptides based on this. And I was talking that why human proteins are better because they are innate part of our body. So you won't get immune response to it. The body will be able to take it if we make drugs out of these proteins. So currently in my lab, my PhD students, they are trying to look into proteins which are similar to transthyretin, such as beta-2 microglobulin. She has found that beta-2 microglobulin, which is present in the kidney, it can also actually inhibit the E. coli. Now she's trying to understand that what is the mechanism, what is the interaction between these two proteins that lead to inhibition. And if it is inhibiting the biofilm, what are the consequences of this inhibition in context of the kidney diseases. Another example where you can use probiotics. So in our gut, we have a lot of probiotics that keep us healthy, that keeps the gut pretty much healthy. So one example is lactobacillus. So what we have done is we have taken lactobacillus supernatant and tried to see whether it has an anti-biofilm activity or not. So it does have anti-biofilm activity. And we are now trying to, so the supernatant is a very complex system which has a lot of metabolites. It has certain toxins also. So now we are trying to figure out that where this supernatant is targeting. And we are also trying to understand is that whether it is affecting at the protein level or it is going down to the genetic level or not. So this is another example where we have used a plant extract, a plant which is very common in the desert area. So we wanted to see whether the plant extract has an anti-biofilm activity or not. So we found that the plant extract doesn't have much anti-biofilm activity. However, when we make the carbon dots out of this plant extract, it enhances its anti-biofilm activity. So we do not see any biofilms when we add carbon dots made from this plant extract. And this work we have done in collaboration with Dr. Viraj in our department. So this shows that certain Ayurvedic preparations or mono-herbal preparations, they might be less effective by their own. But if we can modulate them as nanoparticles or as carbon dots, their efficacy might increase, their stability might increase. So that's where I think our expertise can come into the picture, that we can test them, and it's not just a screening assay. Rather, we are going to test that which one is working in what manner, and how and where it is going to target inside the human body. So that is where I would like to end, and I would like to thank my lab here. So these are the wonderful people who I have. Bharat is working in the Iotech project. So what he has started is he's trying to standardize biofilms from different bacterial species, and we look forward to get certain formulations from the Ayurveda University, and to exactly see whether it is inhibiting the cellulose, and if it is inhibiting the cellulose, where it is going to do the final inhibition, whether it is binding to the extracellular DNA or it is binding to the protein polymers. And if we could figure out, we can have combinations and permutations of these drugs to ultimately target biofilms. And what I have shown you right now are just the monospecies biofilms. However, in nature, we have polymicrobial biofilms. That means biofilms from more than one type of bacterial species. So if we can combine these two different combinations, we might get a better effectiveness against more than one bacterial species. Okay. So with this, I would like to end that the bacteria is one of the primitive organisms, which shows us that there is a strength in the unity. That means if they live together, nobody, even the human beings, can kill them. So and so in the similar manner, if we all come together on the same platform, we hopefully will be able to tackle the vast problem of biofilms. So with this, I would like to thank all of you for listening to me, and I'll be more than happy to take any questions if you have. Thank you.
Thank you, Dr. Sab. It has been a very knowledgeable lecture, and many new things we have gone through. And certainly, this will prove, this will prove better. The students who are present here, the PG scholars who are present here, certainly somewhere in their researches, they will be having a very fruitful solutions from your lecture, as well as if they are having any type of query, they can ask you in this session as well. If there is any query, anybody is interested to ask any question, he is welcome. He or she is welcome. Any question from any side?
May I question, sir?
Please name yourself.
Sir, myself, Ravi Pratap.
Yeah.
I wish to ask.
And your position as well. Where are you working?
Sir, I'm in IIT and working in Iotech Center of Excellence as a research associate. I would like to ask a question to ma'am. That can we find a clear association between biofilms in different organs? Because as per Ayurveda principles, we have Sapta Dhatu, and that's why I wish to find out a correlation among all the organs.
So Ravi, if I understood your question correctly, you are asking for the biofilm connection in the organs of human body, right?
Yes, ma'am.
Yeah. So that's a very nice question, and I think that's where we need to come together and think. So in the literature, what people say that there is an indirect connection. So there might be some biofilm material which might be leaking, and then it might be going to other body parts. So there is no direct connection, as you have mentioned that in Ayurveda also you don't see direct connection, but people are now trying to figure out whether there is actually a direct connection. So as of now, there is no direct connection. There is an indirect connection.
Anybody else?
Hello, sir.
Please.
Sir, myself, Dr. Mukesh Chaudhary, presently working as an assistant professor in HGT University, Gurgaon. Sir, I just
Please.
want to ask one question to ma'am. Is there any role of self-generated alcohols in penetrating the barriers of amyloid and biofilms?
Yes, yes, doctor. So this is a very good question, and
Mukesh.
people have. Sorry, I forgot your name. Uh, so there are reports where people have studied that the self-generated secreted material has some role to play in the formation of biofilms. So it is usually taken care when you have a normal gut. However, in the gut dysbiosis, because of that alcohol or whatever is secreted, it plays a different role. So there is a role of the secreted material in the gut, and that people have recently started to look into it. So the only problem to look into is that people have used mice model because, as you can understand, that human model is difficult to use. So there is some variation between the alcohols that we secrete or we take it out as compared to the ones which are secreted outside the mice. So again, the people have now begun to understand that there is some connection between the gut and other disease, and now people are going back and looking into what all different factors can play into the role, and that is one of the factors. Even there are fatty acids and the metabolites which are generally secreted. They also play a very important role in establishing biofilms. I hope that answered to some extent to your question.
Yes, surely, ma'am. Thank you, ma'am. Thank you.
Yeah, thank you.
Anybody else? It's a very interesting lecture, and we must take full interest on asking the questions. That it shows that you have taken this lecture very much seriously, and it is looking like also, because two of the questions were very much you can say of interest. Anybody else who wants
Hello.
to query? Hello. Welcome, Professor Akashdeep Beshram, sir. He is the professor of Rashna Shari from Parol University, Vadodara. Please, you are welcome, sir. Please
Thank you,
ask your
sir.
query.
And we are honored to listen to such eminent personalities through your lectures. Thank you for the journey of this knowledgeable field, sir. Sir, I want to say one thing that yes, we have got into research and we are deliberately doing some modifications in our research papers, but we are far away from establishing the clinical modes through molecular studies and bioinformatics. So this particular study is very much far from the publications what we see today. Now, if one student, say a research scholar, if he has to study all these through the relation of the active principles and the actions on the proteins, do we have a glossary of the relations which can be established, or if he has to study, ma'am, can you guide on such a glossary which is available for the researchers?
Please call me Neha. Sir, you are asking about the glossary in the Ayurveda or
No,
in
the
connection with
modern
the
aspect
biofilms?
of the relations of the actions on the proteins. Say, if the bacteria and the relations where we can establish the mechanism of biofilms. So, can we can we have a correlative, informative uh, glossary on which, say, somebody is performing uh, a, a research on the action of some drug on liver, and if he has to study what proteins and what mechanism of biochemistry is acted.
That's a fantastic suggestion. To my knowledge, I do not think we have a glossary for that as of now. So what we are trying to do is, so there is a consortia, there is a Biofilm consortium, which is made by India Biofilm Society. I'm a founding member of that. There we are trying to make a peptide library which can act as anti-biofilm agents. But the glossary where let's say in the liver, as you said, that where all the protein and the mechanism is going to work, it is not very clear as of now. In my lab, what we are trying to do is whatever proteins we are studying, and as you can imagine that studying, purifying each protein and working is difficult for the students. So we have just started to work with 4 or 5 proteins, and we are trying to understand the mechanism, and then we are going to reposit it. Of course, we are going to publish it as well, but we are very far from making a glossary as of now. But if more groups come together, then it might be possible maybe after 5 years or so.
Correct, madam. Because see, whenever the research paper is published after establishing a relation, then only we
Yeah.
come to know that yes, this protein
Yes,
acts on that.
exactly,
So
exactly.
unless and until we have a grocery where the researcher can take an advantage prior to
Yes,
going into some
into that
model
particular,
preparation,
yes,
that
yes.
will be help. That will be a big help for the researcher as well. Yes, sir. Thank you for the
Thank you
opportunity,
for
sir.
this session. Dr. Rakesh, I think you are on mute.
Hello, is there anybody asking question? Hello. I think there are no more questions now, but all the questions, whatever have been asked, they were having the gravity in itself. And certainly, if one asks, then that is the query for that is the solution for the other audience as well. So overall, this has been a very nice presentation so far. And somebody is asked, okay. And now I think we are coming to the terminal session of this program. And now I would like to acknowledge for this lecture. I would like to thank our Honorable Vice Chancellor, Sir, Professor Vaidya K. Prajapati, Sir. In the initial stage, he has blessed us by his kind words, by his inspirational words, and hopefully we would be carrying forward this series as well, and we will be having some more lectures the same way in near future also, because we are having so many personalities, but so many authorities in IIT who are having collaboration in with us in through IoTech, and certainly they would be there whenever we call for the lecture in this series. And then I I would like to acknowledge our the pioneer of this lecture series, our Honorable ex-Vice Chancellor, Sir, Professor Dr. Abhimanyu Kumarji. This series was initiated in his time, and I very well remember that very lecture on 15th January when he was there in the initial lecture, and the seed he has planted is becoming big day by day. And I also I would like to acknowledge the director of IIT, Professor Shantanu Chaudhary ji. The same day on 15 January 22, he also blessed us to have this lecture series. And I would like to thank him very much that he has blessed us with such a nice initiative. And now another personality was there, my revered guru, Professor M. Dinakarji from Hyderabad. He has been my guide in Jaipur during my post-graduation, and he has blessed us by being present in this lecture as well. And Professor Dr. Akashdeep Meshram ji from Vadodara, Professor Saroj Patil ji from Pune, Professor Satish Vath from Kurukshetra, Dr. Professor Nidhi Srivastava from Jabalpur, Professor Prem Prakash Vyas, he is the part of us, this Artec Center along with us. He's my colleague here and Dean Research as well here. Dr. Alka Charde from Nagpur, Dr. Mukesh Chaudhary from Gurgaon, and moreover the Dr. Ravi Pratap Singh, and Dr. Amit Gehlot, Dr. Anish Pathak from Bhopal, Dr. Dinesh Sharma ji, Dr. Dinesh Rai ji, Dr. Sanjay Srivastav ji, Dr. Narendra Purohit ji, Dr. Avdesh Handil, and so many others who may be possible. I am not able to name them because I have not seen them. I don't identify them. So whosoever was present in this lecture, I would like to acknowledge him that for his presence. And last and not least, Dr. Neha Janji, because Dr. Sachin Sharma has also been there from Kurukshetra. I'm visible to see him. Okay, Dr. Neha Janji. Because she has spent a very valuable time from his box, and thank you very much for presenting us a very nice and very knowledgeable lecture. Hopefully, Dr. Shambhu, Professor Shambhuji from Kurukshetra. So I'm seeing the people who have blessed us in this lecture. So many people were there. And it is a mini India across here, though we have very selected people, but all the people who were there, they are themselves are the dignitaries in the field of Ayurveda. Thank you very much, Professor Shambhuji. कुछ आप कहना चाहेंगे तो you are welcome, sir. Please, आप कुछ कहना चाहें तो sir.
सभी महानुभावों, आदरणीय प्रजापति सर एवं अग्रज बंधुओं को सादर नमस्कार। और राजस्थान आयुर्वेद विश्वविद्यालय द्वारा जो ये सीरीज पुनः प्रारंभ की, उसके लिए निश्चित बधाई के पात्र हैं। और इससे स्नातकोत्तर अध्येताओं, स्नातक अध्येताओं के साथ साथ शिक्षकों के ज्ञान में भी निश्चित रूप से अभिवृद्धि होती है। उससे सबके लिए आप सब बधाई के पात्र हैं। धन्यवाद।
Thank you for your nice words and for encouraging us. Thank you very much, sir. So I think this way we come to conclude this session. Thank you very much, Dr. Neha ji.
Thank you so much
once again. Thanks once
for your
again.
time.
Hopefully we'll be meeting again, and we'll be there in the IoTech Center time and again there for some sort of researches over there. Thank you very much. Thank you very much. With this we come to an end. Thank you very much. Okay.